I'm going to announce our first speaker. We're thrilled to have Dr. Joe Muenzer with us today to talk to us about potential new therapies. Dr. Muenzer is a professor of pediatrics and a research professor there in University of North Carolina. I know many of you have been out there to visit him, as my family has. He's actively involved in the diagnosis, management, and treatment of patients with MPS and other inborn errors of metabolism. And he's been involved in several of the clinical studies on enzyme replacement therapy. And he's currently a principal investigator for ERT for the intrathecal enzyme therapy. And he's going to share a lot of his information with us, and we're just delighted to have him. Please welcome Joe Muenzer.
Thank you, Steve, for that introduction. It's certainly important for me to note that when I first met your executive director, Barb Whittahay, 20 years ago when I joined the faculty at University of Michigan, there was really no treatments except bone marrow transplantation for MPS I. And today you'll hear me talk virtually of every disorder except for the very rare forms of MPS I. The Sanfilippo C and D, MPS 4B, and then MPS 9, where there's only 4 known patients. I won't mention those, but every other MPS disorder I'll mention. It just shows you how far we've come, but there's still a lot to go, and I'll show you that. So it's going to be a fairly rapid talk. There's lots of things for me to cover, and I'll be clearly available to talk to people afterwards. I hope we'll have a little time at the end to take some questions also. I have lots of disclosure that's required by my university. I currently consult with a number of our biotech pharmaceutical companies. I serve on different advisory boards. I speak for different companies. As you heard, I'm also currently the principal investigator for exciting phase 1/2 intrathecal ERT trial for the severe form of Hunter syndrome. Before I really go into what's happening, I just want to set the stage. It's important for everybody to understand what these disorders are. And so I'm just going to take about 5 minutes, a little more, to really sort of set that stage. So as we all appreciate, the MPSs are a group of rare genetic disorders. There's probably, maybe 2,000, 2,500 total MPS patients in all the US. They're due to deficiency of specific enzymes. And enzymes are really biological catalysts. They help things work. I have some. Thank you, Leslie. Without enzymes, it really couldn't take place. And so what these enzymes are involved in the breakdown or recycling of glycosaminoglycans, which are complex carbohydrate molecules. And they serve all sorts of functions in the body. A lot of the functions we truly don't understand yet, but there's a variety of compounds. Most of them are in the extracellular space, outside the cell, and they have a variety of functions. In the MPS patients, they make these products perfectly normal, but can't break them down due to these missing enzymes. And it's important to note that this recycling doesn't occur outside the cell, doesn't occur in the plasma or serum, but occurs within a particular organelle in the lysosome, called the lysosome. And if you don't get the enzyme and the substrate into that, nothing happens. And this is just an example for those of you who remember your cell biology from high school or college, that most of the cell comprises of other things, but the lysosome normally is a very small component of any given cell, but in the MPS disorder, these lysosomes become filled with undegraded material and therefore can't be recycled, and they accumulate and cause all sorts of tissue and cell dysfunction. So again, these are lysosomal storage disorders. There's now 11 known enzyme deficiencies, 7 different clinical types. As an aggregate, 1 in 25,000 we estimate, maybe more common than that. The hallmark is really the tissue storage of the glycosaminoglycan. That's the first event, but as you'll hear later, there's a lot of other secondary events that we just don't truly understand, and people are now starting to develop therapies for. These are unfortunately progressive disorders that can affect virtually every part of the body, and depending which disorder you can have either physical involvement, neurological involvement, or both, or just bone involvement. And one of the challenges for any therapy is these are very, as I call, heterogeneous disorders. There's lots of different forms even within one type. And I love to show this slide. A lot of the patients I know have known in this slide. Some are from the internet, and just showing you the variability that occurs in this population. And as you can just, if you go into the room where the children are, it's just amazing to see the variability. And for me, as a clinician, it's just always amazing to walk into a meeting where I can see 20 MPS families in one look, children with all sorts of different forms, and it's just remarkable. But this is also the challenge because they're not one disorder, there's a whole series of disorders with all sorts of different severities. Historically, we could always treat mucopolysaccharidoses. My mentor, Liz Neufeld, recognized that cells release small amounts of these enzymes and that it can be taken up by other cells, and she termed this correction factors before she knew these were the missing enzymes. And so we've had this capability, knowledge for a long time, and only now we're starting to really utilize it. One of the keys is that you need tiny amounts of correction, 1 to 2% of residual activity in a given cell, and important in that cell will correct that problem in that cell. The challenge is how to get it to the cell throughout the body, and the bones are one of those challenges you'll hear about in a little bit. I show this slide, that a paper that I wrote with Amy Fisher, who now works for Shire, that in terms of lysosomal enzymes are made in the cell, and they have to get to the lysosome by a particular, what we call receptor-mediated process. But more importantly, if you place the missing enzyme outside the cell with the right recognition markers, it can take it up. And so that's really the uniqueness for these lysosomal storage diseases. I take care of a lot of other genetic disorders, but the lysosomal storage disorders are unique in the fact that putting the missing enzyme outside, the body can then take it where it should be and correct the problem. That doesn't occur for most other genetic disorders I take care of. So what are some of the treatment challenges? As most of you know, or a lot of you know, there's really no effective treatment option for really some of the disorders. For example, we have nothing approved for the Sanfilippo patients. One of the other challenges is why is the central nervous system disease not stabilized in all MPS disorders after hematopoietic stem cell transplantation? That procedure works for Hurler syndrome. It's very successful, but it hasn't been shown the same benefit for MPS II or MPS IIIA and IIIB. It's just unknown why. So we don't have, it's a possible treatment, but it works nowhere near as well in Hurler syndrome. For those of you who are on IV enzyme, it's really a blessing. It dramatically helps, but it requires weekly infusions, and it's incredibly expensive. And in some places in the world, it is so expensive that it's not even approved. And there's some places, even in Canada, where there's some areas historically have not approved enzyme because of the cost factor, because we're talking about from anywhere from $250,000 to $1 million or more of enzyme per patient per year. Incredible, expense. It's clearly worth it, but it's clearly an expense, and we have to probably develop better treatments in the future. One of the big challenges is bone disease. If you walk around and see the patients, a lot of them have abnormal skeletons, and that does not really respond as we currently do ERT or transplantation. Yes, there's some early data that if we treat very young, within the first few months of life, we may alter the bone disease, but that's not been proven on a wide scale. And as we all know, that those of you families have children with neurological or cognitive impairment, we do a very poor job of treating that, except for MPS I Hurler patients. So what are our current treatment options for MPS? It's really, as we alluded to, hematopoietic stem cell transplantation now, either using bone marrow or cord blood as a donor, or IV enzyme to replace the missing enzyme. And here's just a very sort of schematic overview showing that transplantation is clearly beneficial for MPS I in terms of both physical and central nervous system. It's unclear why it doesn't work anywhere near as well for MPS II or MPS IIIA and IIIB. But clearly transplantation does impact the physical disease and stabilizes patients. The problem is that transplantation carries a 5 to 10% mortality at best, and sometimes higher, and therefore limits its use. If transplantation could get to a place of 1 or 2% mortality, then it may become a whole new option for a variety of disorders and may replace enzyme replacement therapy. So now I want to just talk about the status of IV enzyme and then go on to say what's happening new. So to give you what's still old, as some of you are aware, the clinical trial that Emil Kakkis initiated for MPS I started in December of 1997. That's when really began the first intervention for MPSs. You can see MPS 1 enzyme was approved in 2003, 10 years ago now, followed by the MPS 6 enzyme, and then the MPS 2 enzyme approved in 2007. But now we have on the horizon, hopefully, the MPS 4A enzyme that BioMarin is producing. That's going to go to a clinical advisory hearing, so the hopefully is that in sometime in February, I believe, is the date the FDA has to say that they'll hear whether that drug is approved or not. If you talk to some of the families who've been involved in this, they have significant physical improvement, increased endurance, they can do more. This is primarily a skeletal disease, even though this does impact a lot of other parts of the body. And I was others early on didn't expect to see as much benefit as we've seen, and it's been fantastic to see some of these patients do remarkably well. So one hopes that gets approved, but it's a challenge area. It's not a guarantee that enzyme is going to get approved. In terms of other companies now in the field, before there was only the 3 companies, Genzyme, BioMarin involved in MPS 1, BioMarin involved in MPS 6, and Shire Human Genetic Therapy initially as TKT involved in the MPS 2 enzyme. Now there's a company in South Korea called Green Cross who's produced an enzyme for MPS 2. They've used that in patients in Korea, and they're currently looking for worldwide approval, and they're going to try to do clinical trials in the future. So there may be some that product coming online in the future. Ultragenics, a company that Emil Kaka started, is now planning to do the first enzyme replacement trial for MPS 7, a very rare disorder in UK. Hopefully that will happen this year. So that disorder may have some enzyme replacement. That's a disorder very similar to MPS 1 and MPS 2 in terms of physical involvement. In addition to that, there's other companies now who are entering the field and proposing different trials. Signageva Biopharmaceutical has reproduced a recombinant enzyme for Sanfilippo B, and they stated they plan to do a clinical trial in 2014. This most likely will be an intrathecal trial, but I haven't really seen the details yet. But that, again, for Sanfilippo B, there's a clinical trial that's in the planning stages. And that, you can keep track of these in terms of some of these companies have websites and they tell you what's going on. Be aware that these statements are a lot of times very forward-looking, and that you have to be careful what they say on their website, because they're looking to try to get sometimes funding for these, and it's always important to keep that in mind. Another company recently announced, Amicus Therapeutics, that they're developing a preclinical trial for MPS 1 where they're going to take the iduronate enzyme and package it with a chaperone to help stabilize that enzyme, and thereby to decrease the immunogenicity and improve therapy of entering cells. And I'll talk a little bit of that in a later talk, but that's another potential enzyme for MPS 1 that's coming up, potentially to be started in clinical trials in 2014. Remember, once they start clinical trials, it's typically many years, 3 to 5 years realistically, if not longer, before these drugs can ever become clinically approved. But in clinical trials, once patients start, typically they can maintain that particular product. And then the last one I'll also talk about a little later is Armagen is producing a modified human enzyme for MPS 2 and MPS 1. With these enzymes, they're fusion proteins designed to get across the blood-brain barrier, but in theory, they also could treat the physical disease. So there's potentially a third company that could be entering the market in terms of MPS 2, and they're also then planning an MPS 1 clinical trial, and they're envisioning right now to start in 2014 with clinical trials. One of the challenges for all these is there's not a lot of patients out there, and that's going to be the challenge, and people are going to get bombarded looking for candidates, and they're going to have to go to other parts of the world. And so it's going to be a challenging time, but also a very exciting time for families, because there are therapies that will be coming available both on a clinical trial basis and hopefully at some point in the future commercially approved. So now I want to talk about a series of strategies in terms of new therapies and just show you where we are, because there are just lots of things happening. So one of the new treatments I want to talk about is called substrate reduction therapy. And enzyme replacement therapy, as you gather, is aimed to remove the stored material. That is, the enzyme goes into the cell and breaks down what's missing. And that's the ideal treatment if you can get enzyme in there. But what substrate reduction therapy, the goal is to decrease the amount of material that accumulates, and the substrate refers to the stored material. So if you can decrease what's accumulating, therefore you need less enzyme to do what you need to do to break down. And there are some patients who have small amounts of residual enzyme, but it's overwhelmed by the storage capacity. But if you can help that enzyme need to work a little less, you'll get better results for what's there. So that's the concept. The so-called substrate reduction therapy typically uses small molecules. And one of the advantages of a small molecule is that it can penetrate into lots of spaces where a large molecule like an enzyme can't. One of the disadvantages, these substrates that we use for substrate reduction, or compounds we use for substrate reduction therapy, tend to be inhibitors of that enzyme, and that presents a challenge sometimes to how to dose the compound. So in terms of MPS, there's really no approved substrate reduction therapy, no approved medication, but a lot of individuals, particularly in the Sanfilippo community, are on a product called genistein. How many patients' families are using genistein? How many are not using genistein in terms of Sanfilippo's? So some. It's about half. I estimate about half the families in the U.S. are using this genistein. I won't really talk about it extensively, but it's reported in cell cultures to at least inhibit the production or the synthesis of glycosaminoglycans, and then it's been used without any basis in terms of clinical trials that it has any efficacy. It probably is not toxic, even though I can't say absolutely sure of that, and depending on what product used, there may be variable effects, and there's all sorts of different products out there. I know Barbara Burton recently published a paper where she used genistein in a series of patients, open-labeled, and saw no negative effects in that relatively low-dose population. That's published, but this was not a double-blind, placebo-controlled trial. And one of the challenges, as parents know, you want a treatment, so anything you take, you hope that's going to help your child. And we talk about in the research field, placebo effect is that in some areas, a placebo effect, just giving a sugar tablet can have a clinical benefit. And it's important to sort that out from the actual benefit of taking a compound. And that's the challenge of the clinical trials, because when you just take it, nobody knows what's going to happen. I can't predict in any Sanfilippo patient what the clinical course was going to be happening. Some of them get better, some get worse, no matter what we do. And if you happen to take a medication and the child gets better, you can assume that's what happened, even though I can't be sure that wasn't going to happen anyway. I think hopefully we may have an answer in the future that people in the UK, Brian Biggers at Manchester, will actually be starting a human clinical trial for Sanfilippo A and B that will hopefully maybe answer the question of whether this really has any benefit or not. But right now, that's where we stand in terms of genistein. I just want to mention another company that hasn't been as active, or at least as visible lately, but was a year or so ago, which is Zaccaron Pharmaceutical Company, which is bought out by Pfizer, BioMarin. They're developing a small molecule that selectively, what I call, modifies a target glycan. So in this case, what that means is a glycan is a glycosaminoglycan molecules, and they want to actually decrease different types of it, thereby reducing the need for it. And the next slide is an example of that. What happens is that in MPS 2, the ironate 2 sulfatase, the ironate 2 sulfatase enzyme has to remove this sulfate that I'm pointing to there. But if you can develop a drug that prevents the body from ever making or ever placing the 2-sulfate on the glycosaminoglycans, then you never need the MPS 2 enzyme to work. And it turns out this schematic of a glycosaminoglycan chain appears that the 2-sulfate, other sulfated molecules work just as well, since it's not unique to that. So there's an example where if you can modify the body's glycan or glycosamine glycans, how they're made for an MPS II patient, then you may never need any enzyme or very little enzyme. So that's a very novel sort of concept. It's still a long ways away, but that's something that we may see in the future in terms of another type of therapy for MPS II and other forms of MPSs. So that's substrate reduction therapy, primarily with genistein, the one that has taken the center stage. For other lysosomal storage diseases, this is now becoming a very novel and very appropriate topic, and I'm hearing a lot about it at other meetings. So the next topic I want to talk about is called chemical chaperone therapy. How many of you had chaperones growing up? Most of us had chaperones growing up. We have something called biological chaperones. So let me give you a little more biology, a little more science. So proteins, the enzymes we talk about are all proteins. These are chains of amino acids that have to fold in a very unique manner. And they have to fold very precisely for a protein or an enzyme to work. It's been estimated that about 30% of the proteins we all make are not folded properly and get removed out of the cell by a unique mechanism called the ubiquitin proteasome system. The concept is if a protein is misfolded, it gets removed and doesn't get to function at all. And we have biological chaperones to help proteins fold properly. It turns out that if you have what we call a missense mutation, that if you have a little change in the amino acid protein of an enzyme, that enzyme may still work properly, but if it's misfolded, the body removes it. So if we can add a compound to prevent that misfolded protein that is still somewhat active to get to where it should be, then it can work misfolded, but it's got to get to the end result, to the lysosome in terms of MPS II. So that's the concept behind chaperone therapy. So these chaperones typically are small molecules, so again, they have the advantage of potentially getting into the brain depending on the nature of them. They're designed to bind and to stabilize a misfolded protein to get it out of the so-called endoplasmic reticulum space, the part of the cell where this normally gets removed if it's not folded properly, and therefore get it to where it can be in the lysosome. Again, these molecules typically are inhibitors of the enzyme to help stabilize them. And clearly we've seen for other lysosomal storage disease that chaperone therapy has been very beneficial to help improve the quality of treatment potentially for other lysosomal storage disease. So what's the nature of the chaperone therapy in MPS? So again, before I do that, here's just a little schematic. The upper part shows an enzyme. The substrate in this case is the glycosamine glycans the body has. They normally should interact, and then that glycan thing gets cleaved, excuse me. And so that's what should normally happen. If you have a misfolded protein, normally it gets removed from the cell, but now if we add a chemical chaperone, it can bind to this misfolded protein, help stabilize a small amount of it. And remember, only 1 or 2% are needed for normal function. And then this chaperone can come off, and the enzyme now can work in the lysosomal environment where it can be misfolded and still work some. So that's the concept behind chemical chaperone therapy. So a new player in the MPS community is Amicus Therapeutics. They have announced a preclinical chaperone-activated replacement therapy program for MPS I. So what does that mean? So what they're going to do is take a chemical compound that helps stabilize the MPS I, in this case the normal enzyme, and it turns out that even the recombinant enzymes we use for IV therapy are somewhat unstable, even in the infusion bag, and so the concept is that if you add a chemical chaperone to the enzyme in the infusion bag, it'll help stabilize that enzyme while it's circulating and before it gets taken up by cells. Remember, the enzymes, recombinant enzymes therapy only work inside the cell. So they're announcing they're developing this ERT with this human recombinant ironidase and adding this pharmacological chaperone to help stabilize the enzyme. This case is the normal enzyme. and they propose their product would be more stable, less immunogenic, which results in higher circulating enzyme and enhanced treatment effects. So it's a great idea. The question is still in the preclinical stages. They're hoping to start a clinical trial probably potentially next year again. But this is another example of an attempt to help improve and get more mileage out of a very expensive product. And again, it would offer, again, competition. So now here's potentially a competition for the current product for MPS 1. And most of you appreciate probably competition is good because it gives options and forces companies to sort of do their homework and continue to have a best product available. So that's chemical chaperone therapy. I know it's overwhelming. I could take this talk, and I'm going to do it in an hour, and I could easily spend 4 hours. And I know Barb Whittahay didn't want me to bore you for 4 hours, but that's what it takes to really do this. But we're just going to hear an exposure. And I'm just saying this to give you a little break because now I'm going to have you switch gears again and now talk about something completely different that is so-called gene therapy. So what is human gene therapy? So basically, these days, the genetic material is well known and well characterized for all the lysosomal storage disorders, including the mucopolysaccharidosis. The ability to recognize and sequence and purify these different genes for all the MPSs now are readily available. And so human gene therapy is a procedure to reintroduce genetic material. It could be DNA, could be RNA, could be a variety of things, back into a cell, and then that cell now would produce the product that we want to reintroduce. In case of the MPSs, we want to have that cell make the missing enzyme it can't make as well as it should because of genetic reasons. So that's what, in very simplistic form, is human gene therapy. We're not talking about modifying the genome. We're actually putting back into cells a gene that they're missing and have that cell now make that product. It sounds great in principle. And one of the challenges is to carry it out. So there's two ways to try to introduce the genetic material back into the cell. One of those is so-called by physical chemical, just really adding that genetic material to cells and have the cells non-specifically take it up. Not a very efficient process and much harder to control. But the one that's worked most recently and the one that's been used very heavily in the MPS community in animal work is so-called a viral human gene therapy, where we take a modified virus. This virus can enter the cells, deliver its genetic material, that is the missing enzyme that's encoded in that DNA, and therefore then allow that cell to make long-term enzyme. So that's what's been proposed in animals, and I'll show you what's been done in animals very successfully, and I'll talk about what's going to happen in humans. So there's two ways clinically, that is, and the patient, how to give this. We can either directly inject it into tissues, and I'll talk about that briefly in terms of brain injections for Sanfilippo A and B that have started to occur in Europe, or we can inject it IV and hope the enzyme gets to where we want it to throughout the body. Or people can remove cells, transfect the white cells with that, and then hope the white cells are circulating factors. And that's been done in a single patient years ago by Chet Whitley at the University of Minnesota with MPS 2 in an adult that really wasn't successful, but it really opened the door for human gene therapy for MPS disorders. In animals, and I clearly in the past, and I recently closed my research lab, in animals, we can actually effectively deliver the enzyme to the liver and spleen, and there's now evidence showing we can effectively deliver to the brain of animals. So in animals, it's very encouraging studies, and these have been now for the last 5 or 10 years, just more and more evidence that we can correct animals. The challenge now is translate that animal studies into human clinical trials, and that's one of the challenges we have ahead of us. One of the biggest challenges is actually making enough of the gene therapy vectors in a clinical grade material. The FDA requires a very different level of quality control compared to what we do in animals. So that's one of the major challenges, is to make enough of the viruses to be able to use in human clinical trials. As I alluded to, there's been a single adult with MPS II who have received human gene therapy without any toxicity, but without a lot of benefit. There's now 4 French MPS 4A patients who've had direct brain injections. So these patients went under anesthesia, had 6 burr holes placed in the skull, holes in the bone. A needle then was injected into the brain tissue. A little bit of virus was injected, pulled out a little bit, injected some more, and they did that for, so basically 12 injections in the brain. These were amazingly well tolerated, as they have been for other lysosomal storage diseases. All these patients are a year out and have really tolerated the procedure without any really significant negative effects. I think the clinical benefit has really not been reported, but these patients, by and large, were quite severe, and it was initially really a safety trial, and the safety was very successful. So that trial hopefully will continue, and there'll be other patients who will be treated, and we may see some clinical benefit from that. In addition to that, another group in Paris, France, hopefully should have started already, but I think they're going to start this year yet, but it may be early next year, do a similar clinical trial of human gene therapy of direct brain injections for Sanfilippo B. Again, using only French patients by and large. And so that's another clinical trial that's going to happen with MPS IIIB patients. And again, it's going to be primarily a safety trial to show that we can safely do it. And then once you can safely do it, then they'll look for clinical benefit. And there's really 2 big differences. Giving it safely is one thing, but getting clinical benefit is another challenge. And it's important to be aware that in the MPS disorders, there's a certain amount of irreversible disease. So at some point in any patient's development in terms of progressing, that disease may be irreversible. And the goal is to pick patients who we can actually treat and get reversibility or at least stabilization. And that's not clear at what level. And that's one of those challenges. As we all appreciate, the earlier we start these, the better. But we have to show the safety first before we can commence more efficacious trials. Also on the horizon here in the US, Nationwide Children's Hospital has funding to complete the preclinical toxicology requirements prior to starting an intravenous human gene therapy trial. They have evidence in the MPS IIIA and IIIB mouse models that IV enzyme using their particular AAV vector gets into the brain and actually improves the quality and longevity of their mice. So they actually have significant funding to do the clinical trial. They also are in the process of doing a natural history study for Sanfilippo B, their first goal. So for patients with Sanfilippo B who are interested in this clinical trial, you can contact the Nationwide Children's Hospital, and they clearly have a website that you can find them, or through the MPS Society. So then this same group also has funding for MPS IIIA, and that's a little later, but hopefully a few months behind, and so that's going to commence. It's unclear when their clinical trial will start. It's unclear whether it'll start next year or not, but they clearly have the funding and the commitment to do a clinical trial at Nationwide Children's Hospital with IV gene therapy with the hope that the AAV vector crosses into the brain directly. And then I'm also recently aware that in Italy, for those Metabolism A patients, MPS 6, there's a human gene therapy trial that's going to happen there with the idea of just treating the physical disease. And if, in theory, if a human gene therapy trial worked and was successful, you do it once and you never do it again, and the body produces the enzyme instead of having the enzyme produced in a factory, purified, given by IV infusion each week. One of the challenges for these human gene therapy trials is who funds them. These trials are incredibly expensive. They cost a minimum of $2, $3, $4 million to sometimes make the appropriate vector and to do the clinical trial, because the FDA requires safety and efficacy data, and that's one of the challenges for these. It's both the funding and the availability of patients and the appropriate outcome measures to know whether we've done something good or not. So, on to the next topic. Again, I realize we're the incredible amount of material I'm talking about, and I appreciate that. and I realize there's a lot of different aspects we're talking about. You have different levels of your cell biology, but it's important the more we understand the basic biology and hear some of this, the more you can appreciate, because you'll have to make decisions in the future. Do you have your child or yourself involved in a clinical trial, and have to know what is involved? And there's clearly human consent forms these days are really done quite well in the US, but you need to read any consent to know what you're getting involved, what they can and can't do, and what they're proposing to do or not proposing to do. So the next topic I want to talk about is something that I just recently came about, and it's actually potentially exciting work, but who knows how well it'll apply to humans. This is called anti-inflammatory therapy. There's clearly been evidence in the MPS animal models that inflammation is a major component to the disease burden. That is, the stored gags are what occurs early on, but then the stored gags causes inflammation to occur, and that inflammation itself is damaging to the body. And so it's, even though the stored gags started the process, it's once the inflammation gets going, that now causes all sorts of secondary effects. So recently, maybe not so recently, in the last year, Seminaro and co-workers from Mount Sinai recently reported that treatment of MPS 6 rats with a product called pentosan polysulfate resulted in really marked improvement in mobility, grooming behavior, and skeletal disease after the oral administration of the drug. And a lot of you are aware of this. This would not be so remarkable, except this particular product is already approved. This pentose polysulfate is actually an approved drug called Elmiron for the treatment of interstitial cystitis. And so it's one of those, here you have a drug that's approved, and so in theory, a lot of the safety and toxicity has already been done, and now you have to say, can you use this drug for another use? And this is now a big area in medicine that, because it takes so much to get a drug approved, if you have another use for an approved drug, it can really shorten the development time and use of that particular drug. And so this actually was a big issue in the MPS community, because the company, Johnson & Johnson, who markets Elmiron, really wasn't sure what to do. There was an advisory meeting that was called, and I was involved with other MPS people in the country were involved and from Europe to talk about this. But right before that advisory meeting occurred, Mount Sinai School of Medicine and the maker of pentosan polyphosphate, a German company, agreed to collaborate to do a clinical trial. And so that's going to happen. Johnson & Johnson is not going to be involved. It's unclear the details where this clinical trial will occur. Most likely it would probably initially occur in Europe. But here's an example of a treatment of an anti-inflammatory drug already approved in the US and Europe that may potentially have some impact. It'd probably be designed to use with other therapies, since it's not maybe a standalone product. But one of the challenges is that in the animal models, it's my perception there's much more inflammation than in patients. As you know, I'm involved in the intrathecal trial for MPS II, and when we look at the spinal fluid of the MPS II patients as part of the trial, we don't see a lot of evidence of inflammation compared to the animal models where there's much more. So one of the caveats here is there enough inflammation that we will see an improvement in the MPS patients, where clearly in the MPS rat model, there was significant improvement with this oral medication. It's unclear the mechanism and how it works, even though it's proposed as an anti-inflammatory agent. So I just say that's what's out there. A clinical trial needs to be done. It's a medication that's relatively safe. It needs to be done in a double-blind, placebo-controlled manner eventually. But there's another aspect, potentially another treatment that could be on the horizon, and it could potentially be available for all the MPSs, since it's not an enzyme, it's a molecule that would inhibit inflammation. Other people say, why not take other inflammatories? That's one of those challenges. Some of those are potentially have negative effects, but it's one of those areas that we're just beginning to explore clinically. And if this drug has particular success in the MPS patient, it will open up a whole new avenue for other therapies, since there's lots of other anti-inflammatory approved drugs out there that could be tried. The challenge is to do them in a controlled setting and avoid the issue we've seen with genistein, where everybody takes it and we don't know whether it works or not, and it's very difficult to do a trial, for example, with genistein in the US and have naive patients because so many people have been on it. Okay. Almost on time. I know, but I got still a lot to cover, Steve. And I want to leave a little at the end. So the last area that I want to cover is actually intrathecal, how to treat the brain disease in the MPS II. And I want to end up showing some of the results from the clinical trial that I'm involved in. You'll hear this afternoon from Shire from their point of view, but I don't think Shire is allowed to talk about the benefits. But given that I'm not part of the company, I can tell you what I've seen, and I've talked about this at other venues. So as you're all aware that IV enzymes circulate in the blood, and they're taken up by the cells, in the liver and all other tissues. But the brain is protected because of the so-called blood-brain barrier. Prevents us from a lot of things getting into the brain, and that's a good thing because the brain is a much more sensitive environment in terms of toxicity for all sorts of molecules, and a lot of drugs don't cross the blood-brain barrier in addition. And it's unique to the central nervous system. But I want to talk about it also before I talk about the intrathecal, that is putting enzyme directly into the brain, talk about a trial that's going to potentially be coming up in the near future. There's a company called Armagen, and they have proposed a blood-brain barrier penetration of proteins as a treatment for the central nervous system disease. So what are they planning? So this is a company that is planning to do a phase 1/2 trial for MPS II in 2014, and potentially also for MPS I at some point. And they're going to take the MPS II enzyme and fuse it to an immunoglobulin molecule, a monoclonal antibody against the insulin receptor on the blood-brain barrier. So again, they're going to take a protein and modify it, and they're going to try to get the protein across the blood-brain barrier because a lot of molecules can go across through receptors, and they're going to try to do a so-called Trojan horse approach. They're going to try to sneak these proteins into the brain. And they've shown in animals that this works. This technology allows a protein that's re-engineered to cross the blood-brain barrier to get into the brain. And recently in monkey studies, they've very nicely shown that the IV injection of iduronate 2 sulfatase fused to this monoclonal antibody. So the antibody and the protein bind to the blood-brain barrier, and they bind to the insulin receptor that somehow then takes that protein across into the inside, into the spinal fluid, and then now that protein can be distributed throughout the brain. And that happened in these recent monkeys studies, and they found enzyme attached to the fusion protein throughout the brain. So that's actually an exciting possibility. The challenge is, will that enzyme still be active? To my knowledge, they haven't done this actually in disease models yet. So the question is, does the enzyme, once it gets in the brain, active? But it's really another way to get enzyme into the brain. They're looking at all sorts of other disorders with this technology. So we know enzyme will get across. Will enough enzyme get across? And so it's important just to be aware this is in the planning stages. They actually have the funding to do a clinical trial, and hopefully they hope to start next year in 2014. So as you alluded to, the blood-brain barrier, I already said intrathecal, that term you'll now become familiar with in the MPS community, is the direct administration of something into the spinal fluid that occurs around the brain and spinal cord. There's actually been a fair amount of experience to date with intrathecal ERT. The first indications were spinal cord compression due to dura thickening. This is just a MRI, the neck of an MPS I patient, a Hurler-Shea patient. And what you can see is that the dura, the normal covering around the brain, which should be tissue paper thin, it's cardboard, 2, 3, 4 millimeter thick, and literally compresses this spinal cord to the place where the spinal cord doesn't work properly. So one of the indications have been tried is to directly administer the enzyme into the spinal fluid to try to prevent or decrease this compression and avoid surgery, which can be, have a significant mortality in the MPS I patients. Clearly, it's been done. There's been some success in the attenuated forms of MPS I and MPS VI. There's been no major adverse events related directly to giving this enzyme replacement therapy. One of the challenges is for MPS I and VI that the commercial preparations were never made to do intrathecal injection, and you actually have to give the enzyme mixed with artificial CSF to do this, so you're limited with the dose. And again, the overall clinical impression of intrathecal ERT for this indication has ranged from minimal to moderate improvement in the spinal cord compression symptoms. So it's worth trying, but it's still not, probably have to get it earlier the better, and you probably have to prevent it before you really get the maximum benefit. What about intrathecal ERT for MPS I? I think this is actually an exciting area. Historically, we assumed that the patients with Hurler-Scheie and Scheie syndrome had no neurological problem, but it turns out that there are probably significant issues in the Hurler-Scheie in terms of learning and some memory issues that Elsa Shapiro and other people have identified. And so there may be a need for those individuals to have additional enzyme beside the IV. And so there's two trials going on in terms of one, an IT intrathecal trial in correlation with transplantation at the University of Minnesota. So patients undergoing transplantation there with Hurler syndrome can have actually intrathecal enzyme beforehand and then afterwards. So that's an ongoing trial. And then the other exciting one that I've heard some early data on is what Patty Dixon is involved in Harbor UCLA, where intrathecal enzyme is given to MPS I patients greater than 5 years of age or 6 or older who have cognitive impairment. And this is basically using spinal taps to administer that, doing it monthly for 4 doses and every 3 months. And it's clearly one of those patients in the trial has dramatically had benefit with from just personal sort of contact. And so that's going to be exciting, whether that's also going to help some of the neurological problems we've seen in this Hurler-Scheie population in terms of memory and that, whether that's going to be a therapy. And that's an ongoing trial. And it's important to be aware you all should be aware of this website, www.clinicaltrials.gov, because that really has everything that's ongoing in the MPS community. These days, if it's an FDA-approved trial, it has to be listed there, so it's important to keep that in mind. You can see what's going on in the U.S. if you go to that website. So again, www.clinicaltrials.gov should be available to everybody and keep track of what's happening. In terms of Sanfilippo, there's a clinical trial of intrathecal ERT that's ongoing in Europe that's sponsored by Shire HGT. This afternoon you'll hear from Shire and they'll talk about this trial. I won't say any more, except there's a trial that occurred both in Manchester and in the Netherlands, and the drug appears to be safe. We've had a significant issue, as you'll hear about. We used an intrathecal drug delivery device in this trial, as we've done for the intrathecal trial for MPS II. So let's just summarize intrathecal ERT before I talk about the MPS II trial. So it's been used for spinal cord compression in attenuated patients with MPS I and MPS VI. Intrathecal ERT has been used in conjunction with cord blood transplantation in Hurler syndrome at the University of Minnesota. Intrathecal ERT is being used for patients with MPS I with cognitive decline or abnormal cognitive ability. It's been used now in intrathecal for Sanfilippo syndrome A and for the severe form of Hunter syndrome. Just a remarkable list of trials that are happening that 2 or 3 years ago were not available for any of the families. So none of these are clearly approved treatments, but it just shows you what's happening and means to get after another way to treat the brain by putting the missing enzyme directly in the spinal fluid with the idea that the enzyme then can diffuse out through the spinal fluid and enter the brain, throughout the brain, enter cells deep into the brain and actually get stabilization or correction. So now I want to end up very briefly with what I consider really an exciting trial that I've been involved in that, for the last 4 or 5 years, that is intrathecal ERT for the severe form of Hunter syndrome. There's no clearly treatment for the brain disease in the severe form. IV idursulfase, that is Elaprase, at the recommended doses is not expected to cross the blood-brain barrier. Be aware in animal studies, if you take 5 or 10 times the current dose used in humans, there is some evidence that a little bit of enzyme gets across. And that in the future, if recombinant enzyme got a lot cheaper, that may be another way to treat the brain disease by dramatically increasing the cost. But I think if we gave 10 times the dose for the current recombinant enzymes, very quickly insurance companies and other healthcare providers would throw up their hand and say, no way, we're not going to spend $10 million or $5 million per patient per year, given the healthcare costs. So just be aware of that. So preclinical data that Shire has done has shown that intrathecal enzyme can improve the storage in the mouse, MPS 2 mouse that I created years ago, and they also showed in monkey studies that the enzyme, when put into spinal fluid, will actually distribute into deep brain tissue. So we have a clear rationale to use this in humans. The other rationale is in the severe form of Hunter syndrome, we see after age of 2 to 4 years of age, we see progressive IQ loss. And this probably also occurs, if not even steeper, for the Sanfilippo patients A and B. They also have the same neurologic decline. And remember that what's stored in the Hunter brains is the same material, heparan sulfate, that's stored in the Sanfilippo brains of both A, B, C, and D. So the initial trial design for MPS II, he had to be 3 to 8 years of age, had to have some cognitive impairment, but not too much. It was a 6-month open-label extension trial. With an open-label extension trial, we randomized patients to either study drug or no treatment. We initially proposed 3 different doses, 10, 30, and 100 milligrams on a monthly interval, much higher than you can deliver with either MPS 1 or 6. We use this drug delivery device, which is like a portacast. Unfortunately, this device broke because these kids are much more active. They're intended for it, and because of the structural design in the device, it really has not been that useful to administer drug, even though when it works, it really does a nice job, and we can avoid anesthesia in a lumbar puncture. The initial trial design excluded patients with increased intracranial pressure or having shunts, and all the patients had to have 6 months of Elaprase without any significant reactions in the last 6 months. The current design changed because of the difficulty initially of recruiting patients. We went now to 3 to 18 in the trial. They had to have cognitive impairment, but had no lower end of the range, and we also changed our dosing to 10, 30, and we never did the 100 milligram dose because we saw biochemical effects, and we went to a 1 milligram dose to try to get a dose effect. And I'll show you that data in a second. We did cognitive testing at baseline in 6 months, and you'll hear me talk about a GCA, a general cognitive ability score, based on this DAS testing, and that's equivalent to an IQ. It's not exactly an IQ, but you think of it as the GCA is equivalent to an IQ measurement in terms of cognitive ability based on this DAS test. So what's the data show? We enrolled patients 3.5 to 11 years of age. The primary goal was safety, and these patients had a wide range of cognitive abilities. In terms of pre-dosing cognitive assessment scores, we can measure some patients' cognitive abilities, but some we could not measure in terms of this DAS, and some were not measurable, and there was a wide range of different mutations. As we see in all the MPSs, there was not any uniform mutations. In terms of summary results, intrathecal IT, I should say, idursulfase IT that is intrathecally administered was really well tolerated. There was no serious adverse events. I should say this intrathecal drug we used was actually reformulated, so it's the ironate 2-sulfatase enzyme formulated for intrathecal use. It's not the same as Elaprase. Elaprase should never be given intrathecally. We had issues with the drug delivery device, but more importantly, the immunogenic potential of idursulfase IT was very limited. We didn't see any really increase in that, so that really people had antibodies, stayed on antibodies, and they never had any new ones, and all patients continued into the extension study. So the biochemical data that some of you have already heard about, after a single dose, 1 month after a single dose of enzyme intrathecally, the 10 and 30 milligram patients, that is the red and the blue, had an 80 to 90% reduction in their CSF GAGs. So the stored material in the spinal fluid was dramatically reduced. Doesn't say anything about the brain reduction, but the spinal fluid, so it's at least encouraging that the enzyme doesn't work in the spinal fluid, has to go into the cells, and then only then do you reduce GAG storage. And so, but what comes out of the cells in the brain was 80 to 90% reduced and stayed down, where in contrast, the 1 milligram dose had some benefit, but was nowhere near the same, and all the 1 milligram patients after 10 months are now on the 10 milligram dose. This just shows you the cognitive ability in the 10 milligram patients. I want to point out there's 3 that were testable, and in these 3 patients, you can see their baseline over here, excuse me, their baseline over here, pretty much we see in 2 patients, they had stabilization at a relatively good level, and then there's 1 patient who appeared to be stabilized. So in the 10 milligram dose, we had some stabilization of their abilities. In their adaptive behavior score, that also we saw comparable stabilization in the patients who we could test. In one of the 30 milligram patients who we could test, the other 3 we couldn't test, and it's my clinical experience that all the patients that I take care of, which is 10 of them, there's 5 other patients in the UK, that my 10 patients are all clinically stable. They're not getting worse. Some of them are not getting a lot better, but they're not getting worse, and that itself is a benefit. And then in the 1 patient in the 1 milligram dose who we could test with this DOS, really actually lost ability in the 6 months, and that's one of the reasons for going over that 1 milligram patient all went to the 10. So here's the overall GCA score of everybody. Very busy chart, but it just shows you that we're not seeing the same effects that we saw with untreated patients. It's clearly not, they're not normalized, everybody. They're not normal, and some are still significantly impaired. But I want to show you the history of 1 patient. So 1 patient is really the hallmark of this trial for me. He entered the trial at almost 4 years of age, had the mutation predicted to be severe disease, had an older brother die of MPS II at 11 years of age, and he's now been, this is said 37, but it's actually 41 months. And what he's shown is, remarkably, this patient, really stabilization, and not even stabilization, but cognitive improvement. So he entered the trial at 74 as his baseline, and now at 3.5 months later, he has an effective IQ of 94, and this goes across all the domains, verbal, nonverbal, and spatial. It's a single patient, and I acknowledge that, but it shows the potential of putting enzyme in the brain in somebody who's still not too impaired to allow potentially dramatic stabilization. So summary of cognitive abilities, we have testable data on 5 patients, 5 out of 16 yield this cognitive data choice. We're seeing stabilization in patients. The 1 milligram dose experienced cognitive decline, is now receiving 10. And we have correlated data in terms of adaptive behavior, other behaviors that go along with this stabilization we're seeing. So in conclusion for this, the monthly intrathecal administration of idursulfase at 10, 30, and 1 milligrams appears to be safe and well tolerated. The drug delivery device we're currently using does not work well, and we need another one. Cognitive testing in this population is challenging in these severe patients. In the more moderately affected patients for whose sequential cognitive assessments were possible, there's clearly encouraging signs of clinical benefit. This clearly warrants another trial. That trial is in planning stages. If it wasn't for the drug delivery device, we would have already started this trial both in the US and in probably Europe. I want to acknowledge all the people involved in the Hunter intrathecal ERT trial and the sponsor Shire HDT. Thank you for your attention. So we have a time for a few questions. I realize it took a lot of time, but again, I could have spent a lot more time. I think there's some microphones. If somebody wants the first question for me. So what I've said by that, they have the funding to do the clinical trial. But they also have some NIH money to do some of that preclinical data to get that trial going. So that trial is going to happen, it appears to be, without any additional funding. No, funding is a challenge. Thank you. Thank you. Introduce yourself. There's absolutely no question that in the future, most likely we'll be doing multiple therapies. That is, could be gene therapy plus other things, or enzyme plus this and that. I think that's the challenge, how to get two things working, but that's where we're heading. I mean, I can't envision that enzyme itself is beneficial, but there's clearly a lot of limitations to IV enzyme, depending when you start. So yes, I think there will be needs for multiple therapies. How they'll actually develop is going to be challenging. But it's going to happen as people develop different therapies for different disorders. But even though a lot's happening, there's still a lot of need for new therapies. What I say is beneficial, but even with the current enzyme, there's a lot of limitations. For example, the bone disease. There's significant challenges in bone disease. So if we have a treatment for bone disease, as you'll hear about in the next talk, that may be one of those examples of enzyme plus a treatment for bone disease. Thank you. One more question. So the question is, do I see any effects from Obamacare? I don't see effects from Obamacare. I see dramatic effects from the inability of the federal government to get funded and deal with our current budget situation. You know, with the lack of legislative progress in the House and Senate, that's a bigger challenge to research. Obamacare actually is going to be beneficial because there'll be no pre-existing illness issue, and they'll take away the lifetime medical cap. So for this audience, Obamacare, the Affordable Health Care Act, actually should be embraced because it really limits some of these issues that could impact. But research funding at the NIH level is really a significant issue, and that's more nothing to do with the Affordable Health Care Act.